Hello, my name is Astrid Gall. I'm an outreach officer in the Ensemble team, and in this module we are going to have a look at ensemble regulation. Ensemble regulation is concerned with the annotation of the genome with features that may play a role in the transcriptional regulation of genes. We're going to have a look at the data types and the different detection methods that inform our ensemble regulatory build. These are namely epigenetic marks, which come as DNA methylation and histone modification data, transcription factor and RNA polymerase binding sites, as well as areas of open and closed chromatin. Then we're going to have a look at the data that we currently have in Ensemble and that we will have in the future, and how we process it. Okay, I'll start with epigenetics. This is the study of heritable changes in the gene function without any changes in the actual DNA sequence. And these changes can regulate gene expression. Epigenetics data comes as either DNA methylation or histone modifications, and we're going to have a look at both in this module. This is a very old picture here, which I use to illustrate how epigenetic changes can drive cell differentiation. If you imagine a stem cell at the top of this marble run here, running down this marble run and making cell fate choices while doing so, it will end up as one of two terminally differentiated cells. And in fact, if we look at the blood cell line and the hematopoiesis, this is the best example of how epigenetic changes can drive cell differentiation. We start with one stem cell, and that gives rise to all of these other cells in the blood and in the bone marrow, which have the same genome, but a different function, morphology, gene expression, and epigenome. DNA methylation represses the transcription of a gene. So how does it do this? Well, in this picture here, you have in red a gene, and in light blue a promoter region. And in this region, we have methyl groups attached to CG-rich repeats. In this state, the transcription is repressed. However, if the methyl groups are removed, the RNA polymerase and the transcription factors can bind, and transcription can start. We can use bisulfite sequencing to detect where these methyl groups were attached. Bisulfite is a chemical that converts cytosines into uracils, and if we then sequence the resulting DNA and compare it to a reference, we will be able to tell where these methyl groups have been attached, because the methyl groups shields from the bisulfite treatment, and in these areas we still have the C's instead of the U's. The second type of epigenetics data comes from histone modifications. As we all know, the histones are these globular proteins around which the RNA is wrapped. We have histones H2A, B, 3, and 4, which occur as dimers, and they have N-terminal tails. These N-terminal tails can be modified in a variety of ways, starting with methylation and phosphorylation, but there are a lot of other types of modification that are possible too. And we write those down in a certain format, which I've put here at the bottom of the slide. So we start with the histone subunit. In my example, it's H3, so this green protein here. And then we add the amino acid, K36, and then the type of modification, which here is an Me3. That means we have 3 methyl group attached to this amino acid position here. We know that certain types of modifications can either inactivate or activate transcription, and this is what we summarize in the histone code. So for example, shown in green here are the activating modifications, such as H3, K4, ME1, 2, and 3. We also have inactivating modifications, which are shown in red, such as H3, K9, ME2, and 3, and we have some that can do both, and this is shown in yellow here. In addition, there are others where we are not quite sure yet what they do, and these are shown in white. This histone code, of course, is a huge simplification because we have more than one histone per cell, definitely. The next type of data we have that informs our regulatory build comes from transcription factor and RNA polymerase binding. If you go back to our graphic, we have a gene in red, a promoter region in blue, light blue. We all know that the polymerase needs to bind to this promoter for transcription to start, but it can't do it on its own. It needs certain transcription factors to associate with so that the transcription can start. But even that is not enough. We need further transcription factors which are further away, and these are called enhancers. And if those transcription factors bind, it leads to a three-dimensional conformation change in the DNA structure, and that allows the transcription to actually start. So this transcription factor binding to the promoters and enhancers is necessary for transcription, and the combination of epigenetic marks affects the ability and the probability of binding at these sites. There's another sequencing technology here to detect protein binding and histone modifications, and this is called ChIP-seq, chromatin immunoprecipitation. Here we start with a DNA binding protein which is bound to the DNA. We cross-link this to make the bond stronger. We then shear the DNA, and that leads to pieces of DNA still being attached to the proteins. In the next step, we need a monoclonal antibody to pull out the protein with the DNA attached that we're interested in. We then unlink the bond and wash our solution and purify the DNA. We end up with a DNA which was bound to the proteins. And if we then sequence it and compare it to the reference, we will know that these are the areas where protein binding has occurred. The third type of data that we have in Ensemble informing our regulatory build is about areas of open and closed chromatin. So here we have our DNA shown in blue wrapped around the histones in yellow, and you can see there are some areas which are compacted and some which are more open. The areas which are compacted here around the histones are called the areas of closed chromatin, and they are not accessible by the transcription factors and the RNA polymerase. In contrast, the polymerase and the transcription factors can bind to these regions of open chromatin, and this is where transcription starts. So, in summary, this open chromatin is transcriptionally active, while the closed chromatin is inactive. We can use the DNase sensitivity assay to find out about these areas of open and closed chromatin. And the way this works is that we treat our DNA with DNase 1, which cuts the DNA regions in open chromatin regions into pieces. We can then purify it and sequence it, compare it to the reference, and we will be able to tell that these were the pieces of DNA. So this is a sequence where the chromatin was open. This table now summarizes the 3 different detection methods that inform our regulatory build, and the 4 different data types that we have. Currently, our data comes from 3 projects. We have data for human that comes from the ENCODE, Blueprint and Roadmap Epigenomics project, and we have data for mouse that comes from the ENCODE project. We hope that we will be able to include data from farm animals in the near future too. This is from the FANG project, and likewise for zebrafish. All of these projects generate a lot of data for a lot of different cell types, and we cannot show all of this by default. So what we do is we currently show data for 148 human and 81 mouse cell types. We have some criteria for choosing these. So the cell types that we display need to have at a minimum CTCF binding, DNase or FAIR data, and 3 types of histone modifications. If these criteria are met, we display all the transcription factor binding sites and the histone modifications for these cell types. And we predict the activity by processing these data. If you're interested in data for other cell types that we don't have in our browser at the moment, you can add it by using track hubs. Our data processing has 3 steps. We start by taking raw data from a variety of sources. We then predict the positions of the regulatory features, and in the last step we predict the activity of these features in the different cell types. And you are able to view all of this in the genome browser, and that's what we're going to have a look at in a few moments. This little graphic here now illustrates how we process the data in more detail. With our DNA shown in blue, we have raw data signals that come from 3 different transcription factors and 3 histone modification assays. And where these raw data signals reach a certain cutoff, we annotate a peak. So we have 5 peaks here. The peaks are the colored blocks, which come from these 5 raw data signals which have reached certain cutoffs, and another 2 here and 1 here. We then identify patterns in this data, and once we have our patterns, we search for these along the genome. So, for example, we have a pattern that is associated with a known promoter. We will then find the same pattern in other areas of the genome and say, okay, if my pattern is associated with a known promoter, these are quite likely also to be promoters. And we do this for different types of patterns. The next step then is segmentation. This is where we segment our genome into parts which have a regulatory feature, illustrated by the red, yellow and turquoise blocks here, and parts that don't. And these are based on the raw data processing and annotation of the peaks that I was describing earlier. As a next step, we will then annotate MultiCell features. So here we look at our individual cell types, 4 in this example, and for each cell type we will ask the question in a sliding window across the genome, is there any feature annotated in any of these cell types, yes or no? If there's a feature annotated, we will then say, yes, this is a MultiCell feature. And we ask this same question in the next window, is there a feature annotated in any of these cell types? If yes, we annotate MultiCell feature. And this is done along the complete genome. So now we have 3 multi-cell features here. Finally, we will then go back to our individual cell types and ask if these multi-cell features are active or inactive in the individual cell types. So we show the active ones as colored blocks, such as here for cell type 1, and the inactive ones with diagonal lines, such as here for cell type 2 and 3. It's important to note that this regulatory data is relatively complex, as you might have gathered by now, and it's still relatively new. Therefore, we can at the moment not link promoters, enhancers, or any other regulatory features to genes. We show them and you can view them in parallel, but you have to make your own conclusions about how they are linked. For example, we cannot tell that formally that one particular promoter is driving one particular gene. You have to make your own conclusions about this. And likewise, we can at the moment not link regulatory features to gene expression. We do have cell-specific regulation data and also tissue-specific expression data, which you've seen earlier. And again, you have to make your own inferences. This is an area of active development for us. We hope that in the future we will be able to link regulatory features to genes directly. If you wanted to find out more about the ensemble regulatory build, please have a look at these 2 publications here, which you can find on PubMed. What we will do now is we're gonna have a look at the browser, and we will look at a region of the LIMD2 gene and find regulatory features. And we will explore what cell types these regulatory features are active in and what evidence there is to support this. So I'm going to leave my presentation now and open the browser, a new tab in the browser. I will search for the human LIMD2 gene, click Go. And then I follow the link to the location tab here directly in the search results. If you scroll down on this page, which is the region in detail view, you can see that the LIMD2 transcripts are on the reverse strand. And there's a track here which is called the regulatory build. It has a legend associated with it. So we can see a CTCF binding site here, a promoter and a promoter flank. I'm going to click on the promoter, the red block, and I get a pop-up window where I will click on the stable ID, starting with ENSR, to open another tab. This tab is now the regulation tab, and we look at this particular promoter. We are in the summary page now. If you scroll down on this page, you will find a table below this picture here, which tells you that this promoter is predicted to be inactive in 107 of 118 cell lines, and it's repressed in 11. And you can see the details here. If you want to see more detailed information about any cell lines, you can go to details by cell type, the next icon. And on this page, you need to configure your view. So you need to select which cells and which experiments you're interested in. So I will click on this button here, configure cell tissue. And I get a little configuration menu where I can select what I'm interested in. So first and foremost, I need to select my cells. I can do this by either selecting all, going through by alphabet, or using the search box here at the top. I'm going to select CD14 positive monocytes, which are just here, and HeLa cells, which are here. And now I click on the second tab for experiments. And under histones, in the first section here, I will select 3 types of histone modifications, H3K4ME1, 2, and 3. Under open chromatin, I select DNase 1, and in the last section, for transcription factors, I will only select one, which is RAD21. So now that I've made my selection, I need to configure my display. So I click on Configure Track Display. And this gives me a summary of the data that I will show. I can click on cells in this matrix and change what I want to see. And I can also have a look at the summary on the right-hand side. So I can expand sections here to see which cell lines and experiments I've chosen. If I'm happy with everything, I just need to click on View Tracks. And this data loads onto the picture on this page. So my data has loaded onto this picture now. You can see in the addition to the genes and the regulatory build at the top, we now have 2 sections for the CD14 positive monocytes and for the HeLa cells. And for both of these, we show DNase data, DNase 1 data, histone modification data, and in the case of the HeLa cells, also this transcription factors. You can see the raw data signals represented by these colored lines, and then the peaks, which we have annotated where the signals reached certain cutoffs, and the peaks are shown as colored blocks. Looking at this picture, you can already tell that there are striking differences between these 2 cell types. We'll scroll back to the top. If you're interested in where this data actually comes from, you can visit source data, the last icon here. And on this page, you get a table with all the different data that we used and the cell types, and links out to the original sources, ENCODE in this case. If you wanted to explore regulatory features in a wider region of the genome, you can do this on the Location tab. So I'll go to the Location tab now, where we started, and configure the page. Here I choose my regulation menu, and I will go into Features by Cell Tissue first. And here you can see the same configuration menu that you have used on the Regulation tab. And in fact, my cell types and experiments are already pre-selected. What I will add is some methylation data. So I will go into DNA methylation here and select one cell type from this list of cell types. And I will go for Jurkat cells. So I tick the box for the Jurkat cells and close the menu. My data is now loading on the third picture on this page, my highly detailed customizable view of this region. Okay, so my data has now loaded onto this view. I will scroll down to get it into view. We can see below my regulatory build I've got two sections now for the CD14 positive monocytes and for the HeLa cells, which look almost identical to what I've seen on the regulation tab. And I've got an additional track which is called RRBS on Jurkat cells. If I click on that, I will see that the assay here that was used is reduced representation bisulfite sequencing. And for this DNA methylation track, you have a legend, which is shown here. And this ranges from yellow color for 0% methylated reads to 100% methylated reads shown in blue. So you can see for this region that here towards this end of the LIMD2 gene, we have sites with high methylation. Well, this is the end of my demo. There are some exercises in the online course as well, and I hope you join us for the next module.